Hello everyone. In the previous video I have discussed about the genetic code. In this video I am going to discuss about mutations and genetic code. What happens if mutation takes place? What happens in the genetic code? Is there any change taking place or not? So here in this the relationship we are going to find out. Is it right? The relationship between the gene and the DNA can be studied with the help of studying the mutation process. What is the relationship? How is the relationship between the gene and the DNA can be understood best by the study of mutation. Now here when we talk about mutation, we have discussed in the previous chapter about different kind of mutation. Let us say over here insertion and deletion we are talking. So if suppose that a large segment of the nucleotide sequence or the nitrogen bases have been deleted somehow because of the mutation process, then what happens? This can be easily understood. This can be easily understood and detected because this is going to disturb the function of a particular gene. Because in the segment of the DNA where the mutation has taken place due to deletion of the nucleotide or otherwise the nitrogenous bases, so as a result of that there will be a vast change in the characteristic. Some or the other gene is not going to be functional, and as a result of that it can be easily studied or otherwise located, you can say. Now it may be over here due to either loss or gain, I said, either deletion or otherwise insertion. So deletion means loss and insertion is by gain. Extra copy has come, let us say. So in this way what happens? This result mutation, either insertion or deletion is there. So because of that what happens? A gene is either lost or otherwise an extra copy of the gene is gained over here. And due to that it is going to affect onto the particular function the gene was supposed to carry out. When we talk about the effect of a small deletion, here I discussed about what? Deletion, and that also a large deletion here. But now what I'm talking is small deletion. If suppose that a small deletion has taken place, like we had discussed, if you remember, in case of sickle cell anemia in the previous chapter. Sickle cell anemia. In that what happened? At the 6th position of the protein chain, what happens? There is an insertion. There is an insertion of a nucleotide or otherwise nitrogen base. Insertion means over here there is no insertion, actually replacement is there. If you just check over here in the figure down here, for the normal function, the sequence of the nucleotide in the codon is required to be GAG, which is responsible for the coding of glutamic acid, or otherwise you can say glutamate. But what has happened over here? Replacement has been done over here. Replacement has been done. CTC was on the DNA, and due to that what has happened? On the RNA, what has come now? GAG has come. Opposite to C is G, opposite to T is A, and opposite to C is G. So over here GAG is able to code. GAG has come onto the mRNA which codes for glutamic acid or otherwise glutamate. Same thing can be called as glutamate. Now over here what has happened? In place of A, if suppose that T has come, GTG. Now when T is present over here on the DNA, this is DNA at present I am talking. So mRNA that is formed over here is GUG. Instead of GAG, what has come? GUG has come, and this codon codes for valine. So as a result of that, the normal function of HBA gene is changed, and due to that what happens, it has now become what, defective, and due to that what happens over here, the process, the difficulty or the condition that has arisen over here is what, sickle cell anemia. The RBCs have different shape, and due to that their carrying capacity for oxygen is reduced here, and that affects the person in whose body this problem arises or this problem has come. Now coming to what kind of change that we have discussed here. This is actually the point mutation, because there is only insertion or deletion of one single base pair, not pair, only base in the gene, and therefore the mRNA that is produced by that particular gene also will be defected one. So here the effect of point mutation that inserts or deletes a base in structural gene can be better understood by following simple example. Now let us see this example. In this, let us consider that 4 words has been taken, and all 4 words are having 3 letters only, so that we can consider one of these word as one codon, second codon, third codon, and fourth codon. Likewise here, let us see Ram has red cap. Now if suppose that after has, what has happened, B and R, in between that, let us say, what has happened here, B, that is a letter what, a letter B is inserted, a letter B is inserted between this over here, has and B are red, has and red over here. A new or otherwise additional alphabet B has been inserted here in between has and red, and after that if you read, the sentence or the words are going to be changed over here. Let us read that Ram has, and after that what I said here, in between has and red, what is inserted here, B has been inserted. So as a result of that, B, R, E, D is shifted to the next codon. So as a result of that, D, C, A and P is totally separated. It has become only one nucleotide or nitrogen base here. Similarly, when you are going to insert one more letter, say I over here, okay, I after B over here. So as a result of that, B, R, E is there. So B after B, I. So B, I, R, then E, D, C, and then later on along with P, A also has come. Now last A was there, that also has come over here. Further, when you are going to insert, let us say, one more letter, G, one more letter after BI. BI is already here and G is inserted after I. So this is going to become now big. So as a result of that, when you read this, what has come here? Ram has big BIG over here, and then RED, red, and CAP cap. Means Ram has big red cap here. What has happened here? At 3 steps over here. First of all, B was inserted, I was inserted, and then G was inserted. So as a result of that, what has happened? Now the meaning is there that Ram has big red cap. Earlier it was only Ram has red cap. Big was not given here. So big is totally one nucleotide has been added in this case. Same thing we can perform to the another one over here by deletion. Same, Ram has red cap. In that, what is done here? Deletion is done. Insertion was done in the previously. Here we are talking about what? Deletion. Let us say that RED one by one is deleted. First letter R is deleted here. So this is going to be disturbed after has. R is deleted here. So after R, what is there? ED, and then C has been taken from the next nucleotide, next codon. So as a result of that, at the end, AP is left. Second case, R and E both are deleted here. R was already deleted in the first step. Over here E also is deleted. So as a result of that, the codon is what now? Change here. And at the end, what has happened? Slowly, one by one, 3 alphabets, that is R, E and D, are deleted. So as a result of that, totally one letter is missing here. One codon is missing here. And in this, what happened? First one letter, here 2 letters. As a result of that, what has happened here? Shifting has taken place, D, C, A, and then last only P is left. But in the last case, if 3 alphabets are removed, completely one nucleotide is gone out, deleted. Now in this, what conclusion can we draw? Okay, what conclusion we can draw? That means the deletion or insertion, both of them are going to change the sequence of nucleotides. Insertion and or otherwise deletion, when you are doing of one or two bases, what will happen here? The reading frame, over here the reading frame is going to change. Earlier it was there, Ram has red cap. Now over here, after has, wherever the insertion or deletion has been done, is going to change there. Once again, insertion or deletion of one or two bases is going to change the reading frame. And from where it is going to change? The point at which deletion or insertion was done. After that, the reading frame is going to change. And this kind of mutation, in which what is happening? One or two bases are inserted or otherwise deleted. Due to that, other codons are going to be changed now. So this kind of mutation is referred as frame shift insertion or otherwise deletion. Once again, frame shift insertion or deletion mutation means what? Let us separate it totally. Frame shift insertion mutation or otherwise frame shift deletion mutation. If it is inserted, that means what? Frame shift insertion mutation. And if deletion is there, then that means frame shift deletion mutation. Let us take one more example over here. Let us say over here AUG, that is the starting codon or initiation codon. Then next after that CGA, UUA, UAC and triple G over here. Now over here, what has been inserted? One alphabet or one nucleotide has been inserted here. U has been inserted here in between this third and fourth codon. So as a result of that, you see here UAC was there. Now one more U has come to UUA and C is shifted to the next codon. Triple G was over here, CGA. So as a result of that, the amino acid which was to be present or which was supposed to be present at the fourth position, tyrosine, leucine has come here. Over here glycine, in place of that arginine has come. And if deletion takes place at the same position, let us say U is missing from here. Okay, so as a result of deletion, what has come over here? U, A and then over here G has come. What is deleted from here? C has been deleted here. It is little bit colored. UAC was present. C has been deleted. So that's why UA and then this triple G was there. Out of that, one G has shifted here. So as a result of that, UAG is there. And UAG is a stop codon. That means what? When this codon comes onto the mRNA, there is no tRNA or otherwise no amino acid for which this particular codon codes. UAG does not code for any amino acid. So that means the protein synthesis is terminated over here. Protein synthesis stops over here. So as a result of that, if you just imagine the function of or consider the function of this protein and the function that is performed by this. Now this is defected protein, so this protein will not be able to perform or carry out the function that the original protein for which the gene was responsible could have done. Likewise over here also. Now this insertion or deletion, if suppose only one over here we have discussed, here also one by one we have discussed, if suppose that 3 nucleotides are added or otherwise deleted, either inserted or deleted, 3 nucleotide is equal to 1 codon. That means what will happen here? Frame shift is not going to change. Only what is going to change? One of the codon is going to be either deleted or otherwise inserted. Check this point. Insertion or deletion of 3 or its multiple bases, let us say 3, 6, 9 bases are inserted or otherwise deleted. In that case, what will happen here? That let us say that deletion has taken place. So that means one of that particular amino acid for which that particular codon was responsible to code is going to be deleted from there, and the frame is going to remain. After that, whatever amino acid was supposed to be there in the sequence, that is going to be remaining unchanged. If suppose that insertion has taken place, that means what? One amino acid is inserted, and after that all other amino acids are as it is, like how it was before. Now this over here mutation that we have discussed here, this mutation, it forms the genetic basis of the proof that codon is a triplet. What we are talking? This forms the genetic basis of the proof. What proof? That codon is a triplet and it is read unambiguously without any use of punctuations. It is read in a contiguous fashion. 3 codons, 3 nitrogen bases, 1 codon. After that, 3 again, 3 again, 3. It is going to continue till a stop codon comes onto the mRNA. Till then, the new nucleotide, the new amino acids keep on being attached into the polypeptide chain, and the polypeptide chain is going to continue elongation. And when the stop codon arrives onto the mRNA, protein synthesis stops there. So that was about what? The mutation and genetic code. Thank you.